So as I mentioned at the beginning, we have one more speaker in this panel. It's one of those snapshot presentations, and Dr. Amar Sahay from Mass General Hospital, Harvard Medical School, is going to be talking to us. We don't know about what.
Oh, you got it? Oh, sorry. They said it was okay to have extras.
So good afternoon. I'd like to first thank Molly and the other organizers for the invitation. It's a great meeting, and I've certainly learned a lot over the last 48 hours. So as many of you are aware, the adult mammalian brain continues to generate new neurons throughout life. And these dentate granule neurons that arise from stem cells that reside in the subgranular zone of the hippocampus go on to integrate into this circuit. It is now accepted that the human dentate gyrus turns over completely during our lifetime. And a major focus of my lab is understanding the circuit mechanisms by which these adult-born neurons contribute to memory processing in the hippocampus. As many of you are aware, the formation of new episodic memories requires the brain to make distinctions between similar representations. And work by us and by others has found that adult-born neurons play a critical role in the resolution of interference between similar memories. An example of this is parking your car in a different location every day. Consequently, there's interference between the parking lot location today versus that yesterday. And operationally, the hippocampus is thought to perform this process by essentially making two similar inputs more divergent at the level of output, a process referred to as pattern separation. What we and others have found is that if you genetically block neurogenesis, or as we have shown, if you genetically enhance neurogenesis, mice are more efficient in making this distinction between similar contextual representations. More recently, we've begun to think about how these adult-born neurons integrate into the mature hippocampus. And we were inspired by work done by Rusty Gage and colleagues that had found that it suggested that these young adult-born neurons have to compete for perforant path inputs with the pre-existing mature dentate granule neurons. We engineered a genetic strategy by which we could essentially bias the competition between the adult-born neurons and the pre-existing mature dentate granule neurons. To accomplish this, we generated mice in which we could essentially decrease the inputs onto the mature dentate granule neurons and then assess the impact on the competing population of adult-born neurons shown here in red and purple. What we found was that when you genetically decrease the inputs onto the mature dentate granule neurons, you see a dramatic increase in the integration of these competing adult-born neurons. What we also found was that there's a dramatic increase in the activation of these neural stem cells, suggesting that the mature dentate granule neurons here that have lost some of their inputs are communicating distally with these neural stem cells. When we flipped the genetic switch in these mature neurons, everything went back to steady state. So in other words, the competitive advantage that these adult-born neurons had went back to baseline, as did the activation of these neural stem cells. The only thing different about the brains of these mice now was that these additional adult-born neurons that had benefited from the competitive advantage now integrated into the hippocampal circuit. Using this approach, we've been able to rejuvenate the dentate gyrus of 11-month-old animals and 17-month-old animals to restore levels of neurogenesis seen in 4 to 5-month-old mice. We've now begun to think about the mechanisms by which these mature dentate granule neurons affect the adult-born neurons. We think this is through direct competition for synaptic inputs. But these effects on the neural stem cells may be through the release of pro-neurogenic factors. And we've just finished an RNA-seq experiment and identified many pro-neurogenic factors, we think are potentially pro-neurogenic, whose biology is completely unknown. At a behavioral level, enhancing neurogenesis in 17-month-old animals is sufficient to improve the discrimination between similar contexts, such as a context A that's associated with the foot shock versus a similar context B. However, both the aged animals and the adult mice, the 3-month or the 17-month-old animals, can easily distinguish between two very distinct contexts. Again, consistent with the idea that the dentate gyrus is being engaged to a greater extent to make this distinction between the similar contextual representations. However, it's important to show at a neural level that you have mechanisms that support population-based coding that allows for this orthogonalization of inputs. And so we looked at population-based coding in these animals in which we've expanded the population of new neurons. And what we essentially found was that the similar context A and context B are encoded by non-overlapping on neuronal ensembles in the dentate gyrus. Together, these data suggest that improving neurogenesis in adult mice as well as in aged animals is sufficient to improve the discrimination of similar contextual representations, but also at a neural level, improve population-based coding that is thought to support pattern separation. Where are we going with these data? Well, we're based on the proof of concept that stimulating neurogenesis is sufficient to improve this form of memory processing in the aged brain. We now would like to identify compounds that essentially target some of these biological agencies, some of these secreted factors that we've identified, as well as using in vivo calcium imaging using miniaturized endoscopes. These are actual calcium traces. This is taken from a review by Garrett Stuber. We can look at population-based coding in awake behaving animals, and we think that this population-based coding mechanisms in the DG and CA3 are very much analogous to the fMRI readouts obtained by Michaela Gallagher and colleagues in individuals with MCI, as well as in studies done by Michael Yassa and Craig Stark and Arnold Backer in the aged brain. So because this might be a shared circuit-based endophenotype, targeting neurogenesis or by targeting the connectivity of the dentate gyrus CA3 circuit may edify new strategies to improve cognition in the aged brain. Thank you for your time, and I'll be happy to take questions.
Questions? Oh, okay. All right. Okay. So now we can take questions for the whole panel, for any of the speakers that presented their very innovative approaches. So I have a question, Dr. Sahay, about you said you used a genetic switch. Could you say a little bit more about that?
Yeah, so we identified a transcription factor that negatively regulates dendritic spines, and we engineered a mouse model in which we could inducibly upregulate this transcription factor and eliminate dendritic spines only in the mature neurons. So when the spines went down, we found that the competing adult-born neurons benefited in terms of their integration and competition. This mouse model also allowed us to reversibly flip the switch. So when we brought the transcription factor levels back down in the mature neurons, the spines came back. So not only did this tell us that we could reversibly modulate spines in the mature neurons, but it also allowed us to directly assess the competition between the adult-born neurons and the existing mature neurons.
Yes.
I had a question about, oh, sorry.
Go ahead.
So that same study, when you are increasing neurogenesis, you're getting better pattern separation. But it seems like that's a transient effect. What happens at steady state? So once if you are increasing neurogenesis over some long period, how does that influence pattern separation in a long term, or do you have some idea of how that would work?
Yeah, so we haven't done the experiment where we chronically expand the pool of new neurons, and that's because of a limitation in the system. However, I would predict that using another strategy that we've engineered in the past to genetically enhance neurogenesis, that when you push the system, you may elicit homeostatic correction. Right. So, for example, if you enhance the survival of adult-born neurons chronically, it might be that the proliferation of the neural stem cells goes down at some point. What this study told us is that the mature neurons can communicate upstream with the neural stem cells. And so there are likely to be homeostatic mechanisms that will kick in at some point if the manipulation is chronic. But you may not need to chronically expand the population of new neurons. So one of the experiments, and consistent with some of the themes in this meeting about reserve, is that if we expand the population of neurons in middle age, does that confer a cognitive benefit in 17-month-old mice?
Right. So
which we haven't done as yet.
I guess the rest of the question is, if there were an intervention based on this, it would be a static intervention, not some compound that you'd take throughout life.
Yeah, I think that's one way to think about it. Yeah. Yeah.
Yes.
Great talk, Amar. I'm thinking more from a synaptic information processing standpoint. So when you do the switch and you basically decrease the inputs to the mature granule cells, so presumably you would have some compensatory mechanisms in terms of network sparsity, or even the Overstreet actually showed with IBAX mice, right? You got increased LTD. So remind me again, with these mice, in terms of their electrophysiology and processing capability, and specifically because we're talking about pattern separation sparseness, how does that actually change when you switch?
Yeah, so in these animals, we show that when you enhance neurogenesis, in this task where animals are exposed to two different contexts, there's a mismatch-dependent enhancement in sparseness in the DG. So we don't know what the molecular mechanism or the circuit mechanism could be. Cholinergic input onto these new neurons that's telling the neurons that there's a mismatch, because when the animals experience the same context over, we don't see this increase in sparseness in the DG. Regarding the elimination of spines, I mean, our system is reversible, so the spines come back, and we've also been able to dissociate the contribution of reversible elimination of spines from the increase in neurogenesis using a pharmacological agent in the same study. Yeah.
Marcel.
I also had a question about the neural circuitry. The dentate gyrus receives very strong cholinergic input at the inner molecular layer, and there's a lot of evidence about cholinergic influence on experience-induced plasticity. Did you have a chance to look at the influence of cholinergic input in these processes that you described?
No, we haven't done that. The only thing that we've done is we've mapped the inputs onto these adult-born neurons, and they're very clear inputs from the medial septum that project onto these adult-born neurons, but the functional significance of those projections have not been explored.